Hi everyone, it's Dr. LeBlanc with Unit 6 Regulation of Eukaryotic Expression, Epigenetics. Our learning goals are to understand the definition of epigenetics, to understand the environmental factors that can influence changes in the epigenome, to understand the heritability of epigenetic information, to understand how chromatin is remodeled, to understand the modifications to DNA and nucleosomes made to encode this information, to understand how epigenetic modification influences gene expression, and to understand what euchromatin and heterochromatin are, and finally, to understand the process of transcribing DNA that is bound up by nucleosomes. Remember, inside the eukaryotic nucleus, DNA is wrapped around nucleosomes. This is a 30 nanometer fiber, which doesn't look like it has much access for RNA polymerase and all the other transcription factors that need to bind to the DNA in order to accomplish transcription. And indeed, that is the case. When a gene is to be transcribed, it has to undergo changes to make it a more open conformation. And we need to move some of these nucleosomes around. When the DNA is in a more open conformation, it can be transcribed and you can finally end up with an active protein. So epigenetics are heritable changes in gene expression that do not involve changes to the underlying DNA sequence. The sequence of bases in the nucleotides of a gene do not change. However, through epigenetic modulation, epigenetic changes, transcription can be modulated or altered. What are some of the things that can do that? Diet, seasonal correlations. If you get sick, if you do drugs or smoke cigarettes, how secure you are, how much you exercise, which other organisms you share that share your body. If you take medicine or alternative medicines, social interaction, psychological state can all alter gene expression, not by changing the DNA sequence, but by changing some of the other modifications to your genome. What kinds of changes constitute epigenetic modifications? Well, there are two main types: modification to the histone proteins in the nucleosomes, and also methylation of nucleotides. So, methylation of nucleotides can alter gene expression as well as modification to some of the histones in nucleosomes. And we're going to look at both of those types of modifications. An excellent website to further your knowledge about epigenetics is the ENCODE interactive website. You can go to Naturecom ENCODE and learn a whole lot more about this very fascinating aspect of genetic modulation. Two common ways in which chromatin structure is altered are by ATP-dependent chromatin remodeling and by covalent modifications of histones. So a chromosome has different regions of its chromatin that are more tightly or more loosely bound. Euchromatin is the region of the chromatin that is less tightly bound, and heterochromatin is very tightly bound. Heterochromatin can further be divided into facultative heterochromatin and constitutive heterochromatin. Let's talk about this one first. So at the telomeres and centromeres are DNA sequences that are always bound up very tightly into constitutive heterochromatin. Every single chromosome, the telomeres and the centromeres, are always heterochromatin. They're all always on. They are always heterochromatin. Facultative heterochromatin can switch in between euchromatin and facultative heterochromatin. Those are interspersed in regions, other regions of the chromosomes, where you find mostly euchromatin. Facultative heterochromatin means that a gene has been turned on at one point and subsequently turned off. The more open conformations of euchromatin are correlated with higher levels of transcription, whereas the more closed conformations of heterochromatin are correlate with less transcription. So let's look at some of the types of modifications that can happen to chromatin. One of the things is that you can alter nucleosome position. Here you have some tightly packed nucleosomes and some other tightly packed nucleosomes. Well, you can push them aside or just have fewer nucleosomes per length of DNA. Another type of modification is to completely evict a nucleosome. The histone octamers are totally removed and you have this open region of the DNA. Finally, you can often replace variants, histone with other variants. Histones themselves are among the most highly conserved proteins on the planet. The histones in nucleosomes from yeast to humans vary almost not at all in sequence of the amino acids. However, there are certain variants that can crop up at different times or in response to different stimuli. So the standard histones are H1, H2A, H3, H2B, H3, and H4. However, you can have replacement of some of these histones in specific cells. For example, H1 naught variant is associated with more tightly compacted chromatin and gene repression. So in regions where there are facultative heterochromatin and genes have been turned off, you often find the H1 variant. Other types of cells where you need to compact the DNA as tightly as possible and not allow any transcription whatsoever are in sperm cells. So how do we modify the tails of histones to facilitate these compaction or more open conformation of chromatin? There are several different types of residues on the amino terminal tails that protrude from the globular domains of the histones. Each one of the histones has this amino terminal domain protruding, and lysine and serine residues and arginine residues are often the targets for other covalent modifications. Most frequently are acetylations. Here we have lysines that are acetylated. Most of the acetylations occur on lysine residues. You can also have methylation, which can occur on all three different types of residues, the serines, the lysines, and the arginines, and phosphorylation. So all three major types of modification: acetylation, methylation, phosphorylation on the tails. And you can appreciate that there can be a whole variety of modifications at any one nucleosome. So here is a depiction of one nucleosome with tails sticking out. These are only 4. There should be 8 altogether. And you can see all kinds of different modifications that are possible. So a number of different patterns that are possible is very large. One of the most common mechanisms for changing chromatin structure is to acetylate histones. Okay, so that's the one we're going to focus on the most. Histone acetyltransferase is the enzyme that puts acetyl groups onto the amino terminal tails of the histones protruding from the nucleosome. So histone acetyltransferase will go ahead and put acetyl groups on the tails, and histone deacetylase will remove them. What is the effect of acetylation? It is to have the DNA less tightly bound to the histone protein. So acetylation is associated with a more open conformation of chromatin. When you have a more open conformation, you usually are able to transcribe the DNA into RNA. The histone code is the entire pattern of modifications that occur on the genome to the histone tails. Euchromatin, the more open conformation of chromatin, can have one pattern, and heterochromatin will have a different pattern of modifications. Notice also that there are other proteins that are bound here. In an active gene, a gene that is actively going to be transcribed, you often find a nucleosome-free region around the transcriptional start site and then another nucleosome-free region around the transcriptional termination site. Around these nucleosome-free regions, the nucleosomes tend to be precisely positioned, but less so in the middle or outside these nucleosome-free regions. When a gene is silent, there are no transcriptional factors or RNA polymerase bound here, or active RNA polymerase bound. Okay? In order to get activators to bind, the enhancers have to be close to this nucleosome-free region, and that will initiate the process. When an activator binds to an enhancer, it often also binds a complex called SWI/SNF that recruits histone acetyltransferase. This is the enzyme that's going to affect the change in chromatin structure. Okay? Acetyltransferase will start to transfer acetyl groups onto the histones of the nucleosomes just in front of the start site of transcription. The pre-initiation complex will form in this nucleosome-free region, and RNA polymerase will begin to transcribe the gene. How does it do that with all these nucleosomes in a way? Well, the nucleosomes are evicted. The acetylated nucleosomes are removed from the DNA in front of RNA polymerase. Once they are removed, the evicted histones are then deacetylated, and then they reassemble on the DNA behind RNA polymerase. This is a very interesting flash animation with a tutorial at the end to show you how the remodeling of chromatin around the start site of transcription is necessary in order to get a gene transcribed. And this is a yeast model. So in addition to modifications on the histones, you can also modify the DNA directly. And the types of residues that get modified on DNA are called CPG islands. Okay, here is a CPG island. This is a C residue with a phosphate in the middle and then followed by a G residue. So it's 5' C, 3' G. This is the unmethylated state. This is a hemimethylated state where only one of the C residues has a methyl group on it, and this is a fully methylated state where both C residues have the methyl group attached covalently. So what does this mean for a gene if your DNA is methylated? Well, active genes tend to be not methylated. Here's a CPG island here, the CPGs, a few different CPG residues, motifs are found in this region. When it's not methylated, an activator protein will bind to an enhancer and you will get transcription. If the DNA at those CPG islands is methylated, however, activators won't bind and you won't get transcription. So methylation is associated with inhibition of transcription by blocking an activator protein from binding. There's a second way as well. Methyl CPG islands tend to bind a methyl CPG binding protein. Okay, so they won't bind an activator, but they'll bind this protein instead. This protein starts to recruit other proteins such as histone deacetylase, which tends to make the DNA more tightly bound up around nucleosomes. So the methyl CpG binding protein recruits other proteins that change the chromatin into a more closed conformation, which represses transcription. DNA methylation patterns and histone modifications are heritable during cell division. This is important because if you want to affect change, you want to pass that information on from mother to daughter cells during the process of DNA replication. So when DNA is hemimethylated, it recruits proteins that fully methylate the DNA. Okay, this is a replication fork, and we started out with our parental strand being methylated here, and our newly synthesized DNA does not have the methyl group on it yet, but this hemimethylated DNA is the target for enzymes that will put the DNA, put the methyl group on the DNA, and now you have fully methylated DNA. So you can pass this information on from mother to daughter cell. Similarly, histone modifications are also passed along at DNA replication. So after the DNA is replicated and bound up by nucleosomes, during that process, enzymes can come in and methylate the tails of histones to match the methylation pattern on the opposite strands of DNA at the other side of the replication fork. So the enzymes that recognize the hemimethylated or the half methylated or half modified histones are responsible for perpetuating these patterns of DNA methylation and histone modifications during cell division. So in summary, chromatin can be remodeled by removing nucleosomes via ATP-dependent mechanisms and by altering the modifications on histone tails. Histone proteins are among the most highly conserved proteins in eukaryotes. Their amino acid sequences do not vary significantly from yeast to humans. However, histone variants do exist in specialized cells. For example, an H1 that compacts the DNA very tightly is found in sperm. Histones have a short amino terminal tail that protrudes from the assembled nucleosomes. That is the site for several types of modifications, including acetylation, methylation, and phosphorylation. Actively transcribed genes have fewer nucleosomes and a nucleosome-free region at the promoter and 3' end of a gene. During transcription, histones of nucleosomes in front of RNA polymerase are acetylated, evicted, deacetylated, and reassembled on the DNA behind RNA polymerase. Studies in yeast show that active remodeling of chromatin can reveal activator binding sites. Methylated CpG islands are associated with repressed genes. Methylated DNA is associated with shutting down transcription by two separate mechanisms: binding repressors and recruiting other proteins to compact the DNA. Epigenetic information is heritable at cell division, and hemimethylated DNA becomes fully methylated at the pattern of histone modifications is read and duplicated both by special enzymes onto newly assembled nucleosomes after DNA replication.